Hello students, this is a recording for Biology 1406, Chapter 8 in the OpenStax textbook, Photosynthesis. This is the third lecture of Unit 3, the third and final lecture of Unit 3. All right. Photosynthesis, of course, is one of the defining features of autotrophic organisms, and hopefully y'all are all well versed on the difference between autotrophs and heterotrophs. Right? So autotrophic organisms produce their own organic molecules from inorganic precursors. At its very simplest, that's going to be water and carbon dioxide. The carbon dioxide is reduced by taking electrons and hydrogens atoms off of water and adding them to a carbon dioxide molecule to make a COH molecule. All right, making of a sugar or other organic molecules from inorganic precursors is the defining feature of an autotrophic organism. Further subdividing that topic, there are two general ways in which autotrophic organisms function. One, and perhaps the older system, is chemoautotrophs that use a energy source in the form of usually an energy-rich inorganic compound, something that can be oxidized, and that is the power source for making organic molecules from water, carbon, and other mineral elements. So that's chemoautotrophy, and we see organisms like that. They're inevitably bacteria living associated with volcanic vents under at the bottom of the ocean. Mid-Atlantic Rift would be a great example of that. Hot springs, geysers, and places like that where there's a lot of thermal energy available. Photoautotrophs are the other great big camp of autotrophic organism, and by far the most abundant. And all plants, algae, and cyanobacteria are photoautotrophs, and they use the energy present, the kinetic energy present in photons as a power source for generating their organic molecules from inorganic precursors. All right, so a little bit about autotrophs and heterotrophs. So another important idea of photoautotrophy that hopefully is not alien to you is that all food chains, which is an ecological concept, what eats what eats what eats what, food chains and food webs are based on autotrophic organisms. So the base of the food chain always must be an autotrophic organism, either a chemoautotrophic or a photoautotrophic. Down at the bottom of the ocean, there's quite a few chemoautotrophic food chains, or at least partial food chains that are based on that chemoautotrophic bacteria. Those food chains inevitably intersect with food chains that derive their energy from the sun via photosynthesis, but the idea still holds. So, at its simplest, photosynthesis is using the power of sunlight or using sunlight as a power source to push the combination of carbon dioxide and part of water, the hydrogens of water, to form carbohydrates, and then oxygen is released as a waste product, not necessary in the photosynthetic pathway. It's a byproduct. This was an interesting experiment done, I should have the reference, but I don't have it ready. But famously, plants were grown by experimenters in the 1700s even, trying to determine what material it was that the plants consumed to grow. And of course, the reigning theory at that time was, and seemed very logical, was that plants formed themselves from material they drew out of the soil. Very, very reasonable. Well, experiments were done growing plants in a very carefully measured amount of soil for a number of years, and then pulling those plants out of that soil tediously and carefully collecting all of the soil and weighing the plant dry and the soil dry afterwards and comparing. And what they found that was only a tiny, tiny fraction of the dry soil weight was consumed during the course of the plant's growth, and yet the plant represented many thousands or even millions of times the missing mass in the soil. So clearly, the mass of the plants has to come from somewhere else. As it turns out, of course, the mass is primarily composed of cellulose and water, but as far as organic material, it's cellulose and then, of course, all the other organic components of that plant. And those are composed primarily of water and CO2. And of course, CO2 is drawn out of the air, or for aquatic organisms, it's dissolved in water and pulled in through solution. And water, of course, is imported by the roots from the soil, but it's not a permanent component of the soil, not part of the dry weight of the soil. In terrestrial plants, they have an epidermis that allows for gas exchange through the function of specialized pairs of cells called guard cells, which define an opening. When they're in the closed state, there's no gas exchange. When they're in their open state, there is gas exchange. That structure, that opening, is called a stoma. Stomata plural. Very similar to the pupil in your eye. The pupil is actually an opening in the iris. It's equivalent to this open space right here, and it looks black because you're looking into a dark, confined space that's transparent behind there. All right. We'll go ahead and look at this animation real quickly right here. So I'm pausing our recording while I set it up. Notice the stomas start becoming apparent right here as we get closer and closer. There is one stomata, or one stoma of many stomata. Now we're looking at the spongy mesophyll of the leaf. That is the permeable interior space of a leaf, which is air-filled, if you weren't aware of that. We're looking at individual mesophyll cells, just plant cells of the inner structure of the leaf, and we're going to dive down into one in just a moment here. So punching through, we're seeing, the various subcellular structures inside, passing by a mitochondrion right there. The green spherical structures, or oblong structures, are going to be the chloroplasts. So we'll take a closer look at the chloroplast. This is where the green in plants comes from the photopigments in the chloroplasts. So these are the folded compartments. They're a continuous compartment called thylakoids. The stacks are called grana, but the entire thing is a thylakoid. These green pancake-shaped mounds and structures right here, these are all thylakoid folds, collectively referred to as grana. And that's the site of the photosynthetic activity. So we're looking at ATP synthase spinning along right here. We're looking at the proteins of the electron transport chains, and in blue, and the protein complexes that contain chlorophyll, the photosystems 1 and 2, at work right here. The things moving around appear to be ATP and ADP molecules being regenerated. And so one of the great things about this animation, it just shows you that, unlike in the book where we see an ATP synthase, one copy of photosystem 1 and 2 and whatever, there's millions, untold numbers of these things scattered around on the surfaces of those thylakoid membranes inside each chloroplast of the many chloroplasts inside each photosynthetic cell of the many photosynthetic cells inside a plant. Scale is everything. All right, so let's carry on, and thank you, California Academy of Sciences. We're a good friend of mine from graduate school works. All right, you do need to know this general equation. for photosynthesis, and I've talked to you all about this in our lecture session on Thursday. If you run this reaction in the opposite direction, let me see if I can get my marker pen right here. It's not going to cooperate. But if we turn this arrow around and face this in the other direction right here, we would be looking at the oxidation of sugar in the presence of oxygen to produce carbon dioxide and water in a pile of ATP instead of sunlight. So the cellular respiration grand reaction diagram is this in reverse. But this is photosynthesis, and so the power source for the uphill climb of putting carbon dioxide and water together to make sugar with oxygen as a byproduct requires a power source, and that power source is sunlight. So the photosynthetic process involves two major regions. We divide it up into two large portions that we study, the light reactions and the Calvin cycle, also sometimes referred to as the dark reactions. Not that they have to take place in the dark, but they themselves are light independent. So here's a basic schematic relating those two processes to each other. So within a chloroplast, and that's what we're looking at right here, the outer membrane, the inner membrane, and then the membranes of the thylakoids, which is where you'll find all of the molecular equipment for the light reactions found. On those surfaces right there, we're going to use light energy to strip electrons off of water and protons, so hydrogen atoms plus electrons off of water, leaving only water as a byproduct right there. And then those electrons and the protons that went with them are bound to a transport molecule called NADPH. And at the same time, sunlight powers the regeneration of ATP using a structure very similar to what we've learned about already in cellular respiration, the electron transport chain and ATP synthase. Same idea and very similar mechanisms and molecular structures doing roughly the same job on the thylakoid membrane surfaces powered by sunlight, regenerating ATP. And then what happens is the ATP is used as a power source to pay for the transfer of hydrogen and electrons to carbon dioxide to make a carbohydrate, GA3P in this particular case. That is a 3-carbon sugar right there that is produced. Again, we take CO2, we bind it to an intermediary in the Calvin cycle, very similar to the steps in the citric acid cycle. We add hydrogens and electrons to that carbon dioxide using ATP to power the process and generate sugar molecules. And we do this continuously, recursively. And those oxidized NADPHs, which are now NADP, they've been oxidized, they've had electrons pulled off of them. And the discharged adenosine diphosphates plus organic phosphate are regenerated in the light reactions and available to continue powering this process. So you do need to know the terminology of the bits and pieces of the chloroplast right here. So do recall there is a double membrane out here. So that's a phospholipid bilayer. This is another phospholipid bilayer. And then the thylakoid is a phospholipid bilayer that's folded up really in a terrifically convoluted sense, so that these are all really technically extensions of one compartment, just super folded. All right. The fluid filling this area, which would be the cytoplasm of the cell, is the equivalent of the cytoplasm, just has its own special name. In this particular setting, it's referred to as stroma. But for the record, it really is just another version of the idea of cytoplasm. A little bit about light energy. We've talked about this in laboratory, so you're familiar, but we'll review very quickly. You do need to be able to talk about light energy and electromagnetic radiation a little bit. So this diagram down here represents the electromagnetic spectrum, and it's very small right here, but hopefully you'll recognize the visible light spectrum is this small section in the middle right here, which has been amplified in this drawing right here, going from the short wavelength blue or even violet and ultraviolet end of the spectrum, all the way to the long wavelength red end of the spectrum right here. That's the visible spectrum. It becomes invisible off this way to our eyes and invisible off this way. Not because there's no light or energy there, we just don't have photodetectors that are sensitive to those wavelengths at these ends right here. We have no means of capturing that information. The electromagnetic spectrum runs from the X-ray and gamma ray and X-ray and UV wavelengths through visible light down into infrared, far infrared, and radio waves for the longer waves right here. So that's the entire electromagnetic spectrum. All of these are waves that propagate through space at the speed of light, and they have a kinetic component to it. They are waves of energy, but they also have a particulate nature. And the particulate nature, the particles, the photons, can impart kinetic energy on a surface they strike, which is why things heat up when they're hit by light. And that's the basis of the photosynthetic process, is using the kinetic energy of light to excite an atom all the way to the point where we can break an electron free of it, which is called ionizing that atom, and that kind of radiation therefore is referred to as ionizing radiation. All right, red light has very long wavelengths, and that means less peaks going by per second, the equivalent of, more time between impacts of a peak of energy right here. Short wavelength radiation has very, very short wavelengths, and since the waves are moving at the same speed in all cases right here, that means more peaks per second or per microsecond or hour or day or whatever you want to measure, more peaks striking per unit of time right here, transferring more energy. Therefore, blue light and gamma radiation way up here, but blue light in the visible spectrum is more energetic than red light or infrared radiation or radiation. They have less energy that they transfer. So visible light runs from the 700 to the 400 nanometer range of wavelengths right here. And this diagram is reversed from what we almost always see in the books. It doesn't matter, it's still correct. They've just turned it around. So here's the high energy end of the spectrum, here's the low energy end of the spectrum. And you should commit these numbers to memory. 400 is about the maximum frequency, the shortest wavelength light that can be seen by most things and used, and 700 nanometers is the red end of the spectrum right here. There's a number of organisms that can see further down into the infrared spectrum right here. Pit vipers, for instance, can see way down here in the heat range. A lot of insects can see up here in the UV range of the spectrum right here. Mammals and vertebrates, this is about the maximum breadth of their visible range of light that they can see. And importantly, this is the range of light energy that's used by plants. So the key for plants to use light energy is to have what's called a pigment. And these are called pigments because they have color, so it comes out of the art world really. But these are molecules that absorb certain portions of the light spectrum. And so because they absorb certain portions of the light spectrum, those colors that they don't absorb, the portions that they don't absorb, define the color that we see. So chlorophyll famously appears green because it absorbs blue and red light and transmits green and yellow light relatively easily. So we see those colors left over after the other wavelengths have been absorbed by chlorophyll. Chlorophyll A and B, these two molecules right here, differ in functional groups. There's slight differences in the structure of the molecules right here, and that has a big impact on the particular bits of the visible light spectrum that they absorb. Chlorophyll A is the solid line right here, absorbs a little higher up in the blue range of the spectrum, but not as effectively, and then absorbs quite a lot of red light as well. Chlorophyll B, on the other hand, does a much better job of absorbing blue and short green wavelengths of light, and not nearly as much red light. And then finally, the yellow dotted line right here, or the dotted line, represents beta-carotene, a pigment that appears completely red, orangish red, because it absorbs blue and green light, but does not absorb any red light. Here's beta-carotene. And those are the three most common photopigments that we see in plants. There's quite a few others that are responsible for other colors. Examples being lycopene and zeaxanthin, there I said it, which give us the red colors and yellow colors indicated right here. All right, let's go ahead and take a look at this animation.
The process of photosynthesis produces ATP from ADP and PI by using the energy from light to excite electrons that are passed along an electron transport chain. Coupled with the transfer of electrons is the pumping of hydrogen ions and the splitting of water molecules. The following complexes are found in the photosynthesis electron transport chain: photosystem 2, cytochrome B6F, photosystem 1, ferredoxin NADP reductase, and the complex that makes ATP, ATP synthase. In addition to the complexes, 3 mobile carriers are also involved: plastoquinone QB, plastocyanin, and ferredoxin. Other key components include photons, chlorophyll molecules, protons, water, molecular oxygen, NADP, and the electrons to form NADPH, and ADP and PI, which combine to form ATP. Photosynthesis occurs in the chloroplasts of plants and algae. The process is also found in single-cell organisms, such as cyanobacteria, that do not have chloroplasts. Like its mitochondrial counterpart, the chloroplast electron transport chain consists of several protein complexes and mobile electron carriers. First, a photon of light hits a chlorophyll molecule surrounding the photosystem 2 complex. This creates resonance energy that is transferred through neighboring chlorophyll molecules. When this energy reaches the reaction center embedded in photosystem 2, an electron is released. The reaction center chlorophyll contains electrons that can be transferred when excited. 1 photon is needed to excite each of the electrons in this chlorophyll. Once excited, 2 electrons are transferred to plastoquinone QB, the first mobile carrier. In addition to the 2 electrons, QB also picks up 2 protons from the stroma. The 2 electrons lost from photosystem 2 are replaced by the splitting of water molecules. Water splitting also releases hydrogen ions into the lumen. This contributes to a hydrogen ion gradient similar to the one created by mitochondrial electron transport. After 2 water molecules have been split, 1 molecule of molecular oxygen is created. Plastoquinone QB then transfers the 2 electrons to the cytochrome B6F complex. The 2 protons it picked up are released into the lumen. These transfers are coupled with the pumping of 2 more hydrogen ions into the lumen space by cytochrome B6F. The electrons are next transferred to plastocyanin, another mobile carrier. Next, the electrons are transferred from plastocyanin to the photosystem 1 complex. It is here that photons again energize each electron and propel their transfer to ferredoxin. Ferredoxin then transfers the electrons to the ferredoxin NADP reductase, also known as FNR. After 2 electrons are transferred to FNR, NADPH is made by adding the 2 electrons and a hydrogen ion to NADP. The gradient created by the electron transport chain is utilized by ATP synthase to create ATP from ADP and PI. This is similar to the way ATP is synthesized in the mitochondria. ATP, NADPH, and molecular oxygen are the final vital products of photosynthesis.
All right, so that animation and that film summarized, well, actually it gave in great detail the steps in the light reactions, that first half of photosynthesis. So let's review it very quickly. The key components right here are a pile of proteins all mounted in the membrane right here, which contain photopigments, particularly and importantly chlorophyll. There may be other pigments in here as well, which would give this a different color, but this one's indicating it's just chlorophyll A and B, I assume. And that is a photosystem. So this is photosystem, PS, photosystem. Photosystem 2, named that way because it was discovered second. We have an electron transport chain that includes proton pumps. Very similar to cellular respiration electron transport chain. Different specifics, same idea, same function. Photosystem 2, which accepts those electrons, photosystem 1, which accepts those electrons, is called photosystem 1 because it was the first one that was identified and characterized. Re-energizes those electrons using light energy a second time, and transfers those electrons to an electron acceptor, NADP reductase right here, which reduces NADP to NADPH. That's the fate of the electron right there. And this process carries on continuously, powered by light, stripping electrons and protons off of water, producing molecular oxygen as a byproduct, and supplying us, and that is a source of protons and electrons, ultimately to reduce carbon dioxide, and that's going to be, they'll be carried and transferred via NADPH in its reduced form here, carrying the electrons and protons. The proton gradient that was generated by this first electron transport chain between photosystem 2 and photosystem 1, that proton gradient is used to power ATP synthase, which generates a continuous output of reformed ATP from inorganic phosphate and adenosine diphosphate, just like in cellular respiration. Same idea, same basic mechanism. One notable difference is the polarity of this. Protons are pumped out of the mitochondrion and go back down the concentration gradient. In the case of cellular photosynthesis, the protons are pumped into the lumen of the thylakoid stacks, the granule stacks, into that inner lumen right here, and then they diffuse out into the stroma, the cytoplasm of the chloroplast, and as they diffuse out through the ATP synthase complex. Here's just a closer look at that same thing. So photosystem 2, electron transport chain, which includes proton pumps. Photosystem 1, which transfers electrons to a electron acceptor, which in turn and this is a short electron transport chain really, but it transfers electrons to NADP, reducing it to the NADPH state. Both of those photosystems again are powered by light energy. ATP synthase right here is powered by the proton gradient generated by electrons falling down this first electron transport chain. So photosystem 2 is where water is split. Electrons that ultimately end up in sugars and protons that end up in sugars come from water, and this is the source of those. Photosystem 2 takes electrons from the electron transport chain and reboosts them again to a high energy state, and then those electrons are going to be captured and saved in the form of NADPH. Very much like our cellular respiration chapter, I need you to know the term for this complex right here, photosystem 2, where water is split and electrons originate. This electron transport chain, you don't have to memorize the name of the individual proteins here, but the chain includes 2 proton pumps. Photosystem 1, where we reboost electrons to a high energy state using light again. and then the electron transport chain that terminates in NADP reductase, which is simply an enzyme that reduces NADP to NADPH. So this structure and these reactions right here, the light reactions, feed the Calvin cycle by producing a continuous stream of ATP and reducing NADP to NADPH. And those are the two inputs along with CO2 into the Calvin cycle to generate a 3-carbon sugar. All right. So reviewing the steps of the Calvin cycle right here. First, a couple of terms. Fixation refers to the binding of CO2 to the ribulose phosphate molecule right here. This is this 5-carbon molecule right here. So we attach a CO2 to it, temporarily making a 6-carbon molecule, which is immediately split. Again, you don't need to know the names of these intermediates along the way right here, but you do need to know a couple of terms and names. A key one, and is highlighted right here, is rubisco. This is this extremely abundant protein, the most abundant protein by most people's calculations that do this sort of thing on Earth. And it is the enzyme that catalyzes the binding of CO2 to ribulose biphosphate, hence rubisco, because it's named after the substrate it acts on. And then we introduce for one cycle of this reaction, so to produce 1/2 of a glucose, that's a 3-GP molecule right here, it's half of a full glucose molecule. We need 6 ATP, 6 NADHs, another 3 ATP, so a total of 9 ATP, 6 NADPHs, and 3 molecules of CO2. That's the balanced reaction to get one 3-carbon sugar out of this process. So to make a 3-carbon sugar requires 3 CO2s, there's our 3 carbons, requires actually 12 electrons in the form of the 6 NADHs, and is going to require the energetic content of 9 ATP molecules, 6 over here and 3 more over here to power that reaction. So to get an entire molecule of glucose will require 6 CO2, 18 ATP, and 12 NADPH. So there's your balanced reactions. 3 cycles are required to make one of these 3-GP half sugar molecules. So here we are in just a little bit more detail. Again, to make one 3-carbon sugar, we need 3 CO2s coming in, 9 ATP in total, 6 NADPHs in total. To make an entire glucose, this they're saying here 1/2 of the glucose molecule, but if we wanted to make an entire glucose molecule, we'd have to double all of this. So I mentioned this in our lecture session yesterday. Rubisco, this enzyme, this incredibly abundant enzyme in photosynthetic organisms, is the key enzyme in binding CO2 to a carbon chain, and that process is called CO2 fixation. It also, unfortunately, is very good at binding oxygen to that same 5-carbon intermediate in the Calvin cycle reactions. That process is called photorespiration, and it's a malfunction, if you will, of the photosynthetic pathway, and it gives us a bit of an insight into the age of the system. So the photosynthetic pathways evolved on Earth prior to there being an oxygen-rich environment. In fact, the oxygen released by photosynthesis is the reason why we have oxygen present in the atmosphere and dissolved in seawater and fresh water, of course. And this system, the enzymes and the entire pathway, evolved in the absence of atmospheric oxygen. But as that oxygen became ever higher and higher in concentration, we start seeing an increase in a malfunction called photorespiration. As a result, quite a lot of plants, including this cactus right here, and grasses, and any number of plants that we rely on or that are familiar with that grow in extremely hot and dry environments, have mechanisms to reduce this particular problem right here by basically amplifying or protecting rubisco's exposure, amplifying its exposure to CO2 or reducing its exposure to oxygen, so that the photorespiratory pathway does not accidentally kick into gear. All right, let's see. We're going to go ahead and take a look at this last animation here, and then that'll be the end of this recording.
Photosynthesis is the basis for all life. Thanks to photosynthesis, plants could grow and evolve, and it has served as the foundation for other living beings such as the dinosaurs and the human species.
Oh, that's pretty silly.
Photosynthesis takes place in leaves of flowers, bushes, and trees. First, let's find out what a leaf looks like. On top, we can see a thin layer of wax called cuticula. It minimizes water loss and increases the stability and resistance of the epidermis. The main task of the epidermis is the protection of the leaf cells. Cuticula and epidermis can be found on both sides of the leaf. On top of the epidermis, in the bottom, is a spongy tissue. It allows the interchange of gases for photosynthesis.
It's the spongy mesophyll.
Furthermore, vascular bundles, which can often be seen with the naked eye, allow the transportation of water, minerals, and other products. The palisade cells can be found on top of these veins. Palisade cells contain many chloroplasts that can be seen here as green dots. Chloroplasts absorb a major portion of the light energy used for photosynthesis. So let's
notice the great big central vacuole in this plant cell right here. So here's the cytoplasm around the perimeter. Here's the cell membrane, and they haven't shown it, but a cell wall outside of that. Here's the membrane of the central vacuole full of a salty, sugary liquid substance right here. And then the cytoplasm of the cell with nucleus, Golgi apparatus, endoplasmic reticulum, mitochondria, and all of that, and most notably lots and lots of chloroplasts. Try and remember back to our osmosis lab, one of the last labs we did, where we, as a side project, took a look at Elodea leaf tips from an aquatic plant that we put under the microscope and challenged with salt water and distilled water.
For photosynthesis. So let's have a look at one of them. Chloroplasts have a protective membrane. A liquid called stroma is found inside the chloroplast. Thylakoids are embedded in this liquid. Thylakoids consist of a thylakoid lumen surrounded by a membrane where photosynthesis takes place.
Lumen is a term you'll see a lot in biology and in the sciences. It refers to the space enclosed by something. So the lumen of a cell is simply the space enclosed by the cell membrane. The lumen of the nucleus is the space enclosed by the cell, by the nuclear membrane, etc., etc. So lumen of the thylakoid. Lumen is kind of a standard word that you want to know. The lumen of the endoplasmic reticulum is the space enclosed by the ER, etc., etc.
If we take a close look at the thylakoid membrane, we see the photosystem too. It is linked to the stroma and the lumen. PS2 consists of light harvesting complexes that surround a photosynthetic reaction center to focus energy. Light harvesting complexes consist of chlorophyll, such as chlorophyll A or chlorophyll B. These chlorophylls absorb light, namely photons, and transfer the energy to the photosynthetic reaction center. This is the reason why plants are green, as light with a wavelength of about 500 to 600 isn't absorbed but reflected. This is called green gap. The energy of the photons cause the chlorophyll molecule to enter an excited state, and electrons are ejected from the molecule. As a consequence, the molecule is charged positively. To absorb other photons, the molecule must return to its initial state. Therefore, it is in need of electrons. To get them, the PS2 uses water. Here, electrons are ejected from H atoms, and the H atoms are separated from the O atom. This happens several times, and, as a result, two O atoms combine to form an oxygen molecule that will be emitted later by the leaf. Simultaneously, the positively charged hydrogen ions get into the lumen. The electrons are transported to the PS1 via an electron transfer chain. In the course of this electron transport, protons are being created that get into the lumen. This will result in a protein gradient between the lumen and the stroma, generating a force called proton motive force.
Those protons don't get into the lumen, they're pumped into the lumen by transport proteins that are powered by electrons passing through them.
Proton motive force drives ATP synthase that produces ATP from ADP and a phosphate group. This process is called oxidative phosphorylation. The electrons that
So that's pretty good right there. So yellow represents the protons that are driving the process. Green and blue here represent ADP plus phosphate, and orange, as it's about to be labeled here, ATP, the finished product.
process is called oxidative phosphorylation. The electrons that are transported from PS2 to PS1 are used to balance the loss of electrons in PS1 because PS1 absorbs light, and this results in the oxidation of PS1 and the release of electrons. Electrons coming from PS1 are transported through an electron transport chain to their final electron adapter, NAPD, creating NADPH. ATP.
So remember, it's easy to distinguish between NADH and NADPH. NADH is the reduced electron carrier 1 of 2, along with FADH, in cellular respiration. NADPH is the 1 electron carrier that we talk about in photosynthesis. The way to remember that is that there's a P here for photosynthesis. That's not what it's here for, it's for phosphate, but you can use that as a memory tool. NADPH, photosynthesis. NADH, cellular respiration. NADP, photosynthesis. NAD, cellular respiration. All right?
And NADPH, which are products of this process, can then be found in the stroma, where a process known as Calvin cycle takes place. No light is necessary for the Calvin cycle. During the Calvin cycle, carbon dioxide is reduced to form carbohydrate in a series of reactions. Here, NADPH is the reducing power source, whereas ATP is the energy source. Glucose is the primary product. It serves as an energy source for the plant itself. In the forms of polymers, such as starch, it is also an energy source for animals and human beings. By producing oxygen and capturing energy from inorganic compounds to produce organic compounds, plants are considered to be a very important part of nature and life.
Alrighty, well that's the end of this recorded lecture on photosynthesis. Be sure to check out the recorded lecture on cellular respiration that precedes it, and refer back to your notes from our live lecture on metabolism before the spring break.